Next, we'll look at how the three pathways of lipid transport all link together and interact with one another. So looking at it from a liver-centric point of view, you have the exogenous pathway by which lipids obtained from the diet are packaged into chylomicrons. They are then exported into circulation, travel through the periphery, and are acted upon by lipoprotein lipase, removing triglyceride, producing a triglyceride-poor chylomicron remnant, which is picked up by specific receptors in the liver. Then we have the endogenous pathway of lipid transport, where we have the production of VLDL, which is also triglyceride-rich, containing cholesterol ester as well, but this triglyceride and cholesterol ester is from endogenous sources, such as from adipose storage or those produced within the liver itself. This pathway, similar to the exogenous pathway, the delipidation by lipoprotein lipase and the removal of triglyceride from the VLDL as it converts to an IDL particle. This particle can then be picked up by the liver by receptors recognizing the ApoE protein. However, once the IDL loses this particular apolipoprotein, it then transitions to an LDL particle, which can either interact with VLDL to receive triglyceride at the expense or exchange of cholesterol ester via the enzyme CETP, or it may remain within circulation, being either modified or converted to a small dense form of LDL. LDL also has the potential to be taken up by non-receptor mediated uptake at other tissues and cells. Then we have the reverse cholesterol transport pathway, which links in very tightly with the endogenous pathway in that we have the production of nascent HDL by various tissues, which then is capable of recognizing transport proteins expressed on the surface of cells, which require the removal or efflux of cholesterol. These include ABCA1, ABCG1. The transport of free cholesterol and storage as cholesterol ester is mediated by LCAT activity. Then lipid-rich HDL particles are capable of transferring cholesterol ester to VLDL particles, in return receiving triglyceride, or they can be taken up by the liver via the SRB1 receptor. HDL particles also interact with macrophage and foam cells, which have subsequently endocytized small, dense, or modified LDLs, becoming lipid-rich and potentially atherosclerotic, thereby reducing this atherosclerotic risk by reducing the amount of lipid present in these cells. Then the HDL again can be taken up by the SRB1 pathway. This diagram gives you a discrete summary of the endogenous and exogenous lipid transport pathways and how they interact with the reverse cholesterol transport pathway.